At its most basic level, cancer is a disease of uncontrolled cellular proliferation. Basically, cells begin to grow and proliferate when and where they're not supposed to. And that leads to the development of a tumor. In my lab, I study how cells rewire their metabolism in order to accomplish this task. When you really think about what cells are made of and what making two cells from one cell requires, cells are really made of a lot of proteins, DNA and RNA, and membranes, which themselves are made up of amino acids, nucleotides, and lipids. And these are all metabolites. That's not to mention all of the energy that is required to put it all together into the shape of a cell. So cellular proliferation really is a huge metabolic job. In my lab, we try and understand how that works and develop new ways of disrupting that can be potential improvements to cancer therapy. My name is John Coloff. I'm an assistant professor in the Department of Physiology and Biophysics at the University of Illinois in Chicago. I'm also a member of the University of Illinois Cancer Center. One of the reasons why cancer is so difficult to treat is because it's our own cells that just get a little bit messed up and begin to proliferate. So identifying differences between tumor and normal tissues that can be targeted to specifically kill the cancer cells is very difficult. We've actually known for quite a while that there are big metabolic differences between tumor and normal tissues. In fact, over almost 100 years now. And some of the first chemotherapies were called anti-metabolites because they disrupted this metabolism. In addition, we have imaging techniques that are used to diagnose tumors in patients that image the difference in metabolism between tumor and normal tissues. In my lab, we're interested in understanding the extent of the metabolic changes that occur in cancer tissues and developing new ways of targeting this change of metabolism for therapy. By better understanding the unique metabolic features of tumors, we will be able to potentially develop new drugs that target different metabolic enzymes that could be used to specifically target cancer cells. We might be able to manipulate our systemic metabolism, for example, through changing our diets to improve the ways that existing therapies actually work.